introduction. Yeah. So what I will do in the upcoming like 45-50 minutes is show you some examples, but also allow you to interact a bit with me through a Mentimeter link. I will copy paste the link already in the chat. You don't need to click on it right away, but I'll ask you to use it when I show the Mentimeter interface. Okay, start sharing. Just a bit of some introductional elements. So it is about image analysis, but I actually am also going to zoom in on the challenges of root phenotyping, because it's simply not an easy topic. And I will also explain a bit about the quest that we have been on to enable people to do all kinds of experiments, and often they are connected to measuring the effects of climate change on our food system, on our crops. Just to introduce myself, but on one slide. So it's all about high throughput phenotyping. That's quite a popular term, I think, these days. And I would like to illustrate it with this picture. So it is a field filled with lettuce plants. You can see many plots. Each plot is a different variety. This is a breeding location, so another food production location. And the essence here is that we would like to figure out which plants do best on this specific site, in this specific soil type, in this specific region of the world where there is a certain climate. And you can do this manually, like these people are doing back there. And we are introducing technology to help them speed up the evaluation process. So this is one of the devices that we have to scan the plants, measure how green they are, how tall they are, and just score which plots do best without going in too much detail. And of course, drones are very popular outdoor. But coming back to the topic of today, the roots are hidden. Right? You can't see them, and it's really complicated to measure roots in this situation. So there are actually no tools out there that can really objectively measure what these roots are doing without introducing all kinds of fancy, complicated, or expensive, or even invasive tools. So that's the quest of today. But I'm Rick van der Zedde, indeed, working at Wageningen University in research on plant phenotyping. That's the only topic that I'm interested in, you could say. Doing it as a project manager in the phenotyping center, but also as a senior scientist. I'm running all kinds of research projects. I'm also in the international plant phenotyping network. I have a background in AI. I'll speed up a bit because I've got many things I would like to share today with you. So phenotyping is all about gathering data. Drones capture data, cameras capture data, but it doesn't end there. So, you can capture data by using your mobile phone, taking a picture of something, but then you only have one big blob of data. It actually becomes useful if you turn it into a graph or into a table, and then understand why certain plants grow weak, other plants grow strong. No, the phenotyping doesn't end at the imaging. It actually starts at the imaging. It ends when you have a number, a graph, a comparison between your treatment and your control. And the roots are complicated. And it all matters. So everything matters. So you're measuring a certain phenotype. So a number of leaves, the greenness, ground coverage. So those are phenotypic traits. But then you need to be aware that you also have differences in your phenotype caused by, on the one hand, the variety, the genotype that you have selected in your trial, but definitely also your environment. So where is it growing, but also how much nitrogen did you put on that plant? What kind of watering regime did you decide to use? What's the temperature, light levels? Everything matters, and that results into some kind of a plant. Actually, a plant that hopefully is uniform within its batch, and you can find differences between genotypes or between cultivation practices. That's the environment. So we use this graph as to explain the challenge. So you need to monitor actually all elements. So you need to monitor the DNA profile of your genotype. You need to monitor your environment with sensors. You need to monitor your phenotype. And it can be quite complicated and also expensive. And we asked for money from our government to enable us to do this on a large scale. And that is this MPEG. And it's really needed. So hopefully today also we can actually close the gap, because there's a lot of technology out there. We have been introducing technology to farmers, to growers, to researchers. But the fascinating but also complicated thing is that there are always a few people that jump on the technology right away. A few follow quickly, but then the biggest majority of the users that could benefit from these tools are waiting, are not sure if it works. They might find it too complicated, too expensive. And this gap needs to be closed, because the real impact of all the tools will be generated when many people use the tools. That's my focus, how to close the gap. And that's also something that I would like to ask from you. So actually, if you look at the gap, people that are working within technology quickly jump on new technology. But actually people from biology, they find it maybe more complicated, but it's not their daily work, right? Working with AI or with novel sensors. And if you have enough data, then you can do data science with it. I'm going to ask you to go to the link in the chat and go to the Mentimeter. And I saw, good, some of you already filled it in. I got only a few questions for you. But first of all, I would like to know who has been in the Netherlands, who has been in Wageningen, who has been really on our field station, and who has been really inside one of the MPEG facilities. And just fill it in. At least it gives me an impression of who am I talking to. Good. I thought there were around 22 people in the room, so I'll wait a bit longer. But it's
this has been added to 44 now, Rick.
Oh, then I wait a little bit longer even. Yeah, so this is also a bit of a test question to enable you to use Menti. So you can click on the link on the one hand. If the link doesn't work for you for whatever reason, you could also go to menti.com and then fill in this code. I got a few more questions for you, and this is this is not the most essential question. So please use it. I'll give you a few more minutes, and then I'll jump to the next question, which is actually a real question I would like to have an answer on. Not so many are jumping. Yeah, can you fill it in? Otherwise, I'll go to the next question, and then I'm gonna wait there until people manage to sign in. Here you go. So can you fill in who you are? Because it helps me actually to understand if you have a preference to learn more about technology, or that you are a biology, that is a biologist, maybe a bit new to imaging challenges. You can also freely go actually to the other questions. So if you are in need of more answers or questions, then go to the next slide.
Okay, and just to add again what Rick said, if you have a cell phone available, you cannot do at the same time on your computer, menti.com and enter the use code, then you can enter these questions as well. So you can work simultaneously on your cell phone if you like. Menti.com and enter the use code, and the questions will appear. Yeah,
good, it works. You can do it yourself. We can go back to this. I got a few more questions, but I'll wait and I'll allow you to struggle with the Menti links. But I would like to dive into the root phenotyping challenge, because so the roots are hidden. So somehow you need to measure these roots, and there are all kinds of tools out there. And the most logical first idea would be to use X-ray, although X-ray is expensive and complicated. So I would like to share only a brief example. We have been working on root phenotyping using X-ray with a simple machine, and then in a pot. So that's already one drawback. You can't do it on the field. You need to bring the pot to your X-ray device, and you can actually generate a beautiful 3D reconstructed root system of, in this case, an ornamental plant, an orchid. And it's ideal, you could say. It's non-destructive, it can be extremely accurate, you can see all details, but there's a but. There are many buts actually in my talk. The but is if there's a lot of water in your pot, the contrast between root and watery soil is poor. So this is a super dry pot. The plant is actually suffering from drought, but the result is beautiful. And we did this on a larger scale, also on cucumber plants. And then water was again an issue. So what we did actually, we placed this rock wool block, a common practice in horticulture in the Netherlands, where the water is actually soaked up and the roots can reach the water through this rock wool. If you place them on top of each other, they actually start sucking the water out of the upper block. And when the water is gone, you can make beautiful X-ray images of your root system. So it's a simple trick, takes around 10 minutes for each individual plant. So it's quite some manual work, but the result is beautiful, right? But this root system has been polished manually by a colleague of mine. It took him hours actually to create this. And we are looking at a block of 10 by 10 by 10 centimeters where the root of the cucumber plant is somehow distributed. And then he made an analysis to turn it into, so this is a bit of computer vision, into a skeleton where he could keep track of the root tips, root depth, root density and thickness. So still quite promising, you could say. And then he turned it into numbers. So you could say, okay, there is a root density linked to depth. So most of the roots were actually at 3 centimeters. And then he was calculating that there was a certain radius mean, a certain total volume and total length, and a distribution of volume and length. And these are the numbers that we are actually looking for. And another visualization, also this is a graph of your root length, actually basically looking down how it is organized in 3D, but then flattened into 2D. And I'm going to show you many more examples. So let's stop here because it is nice, but it took 20 minutes to scan the pot by turning it around in the X-ray system. The draining took also 10 minutes. So we are already 30 minutes for each individual plants. The roots got super dry. That's why the contrast is so beautiful. And the plant actually suffered. So our experiment was flawed by the need to have a completely dry root system. And then this manual polishing of the data took too long. It took several hours for each individual scan. So basically the short conclusion is it is not high throughput, it's not fully automated, and it's not scalable towards breeding application, because then you need many, many more plants and it will take you weeks. And that's the whole point. So within MPAC, we have been collecting data, but then really large scale data from fields, from greenhouses, from climate rooms, and I'll show you some examples. And the essence is basically what I showed in the slides before. So you acquire your X-ray or your picture, you organize it, you analyze it, and you visualize it in graphs. So that's constantly coming back in the upcoming slides. And the process is always to go from raw data to information and then make that accessible for anybody interested. International people, companies, and colleagues, researchers in the Netherlands. Okay. Just to illustrate, NPAC is a big investment scheme that is paid by our ministry through our Dutch science organization, and it allowed us to buy very expensive equipment. And you can use it. Anybody could use it because we're open for access. So you can come over to work with us to learn how we are doing. PHENOWEX is exactly the project that we were aiming for. So we had the unique opportunity to buy this equipment. We would like to share this with anybody interested, and definitely people connected to PHENOWEX will learn from our experiences, but also our pitfalls and dilemmas. And when I talk about we, it's Wageningen University in research in this case, but also Utrecht University nearby Wageningen in the Netherlands. Yeah. And if you want to know more, I'm quite active on LinkedIn, so you can connect with me, and once in a while you get pictures like this that illustrate what we are doing. The focus will be on roots, but if I'm going to show a bit of the content of the whole of MPEG, so we have ecotrons, we have root phenotyping systems that focus on plant microbe interaction. I'll show an example right after this. This is all small scale, controlled environments. And the higher we go into the numbers, the larger the scale, and also the less controlled the environment is. So this is a greenhouse where the sun is affecting the plants. This is all indoor. And on the fields, of course, we are completely out in the open. When it rains, plants get wet. When it's, for instance, when you're trying to do a drought experiment, like we did in the last summer, but it's raining all summer, your experiment fails. So that's why we also often do drought experiments in our greenhouse or even in the climate rooms. Then you have control over your environment. Yeah. Okay. So I'll just show you a short video of the 3 modules in Utrecht. So these are the ecotrons. Hopefully the bandwidth is good enough to at least show you a few images. So these are big cylinders where the roots are growing in interaction with other plant species. There are a few cameras, but mainly sensors in there to monitor the fluids inside the soil, which are then sequenced to figure out which bacteria are active around the root systems. So it's like a tiny field, you could say. This one I'll zoom in a bit more. So this is a small chamber where there are agar plates automatically filled and also sown with seeds. Where the first germination effects of your root is visualized by imaging. And when all kind of treatments can be added, like this one, figure out how these roots actually evolve over time and how different treatments and controls are measured by cameras. So it's definitely image driven. And that's called Hades. So I'm going to focus on Hades. Hades has a robot system to move these agar plates around. He has got imaging boxes to take images of these agar plates in which the roots are growing. And then we have multiple imaging systems. So then there's a hyperspectral camera, chlorophyll fluorescence camera, and an RGB camera. I would like to show you some simple results. So it's a small agar plate. We have the roots that germinate. We can see a bit of the shoot. We can see a bit of the roots. And this is the raw data. And then there is some algorithm. So this is where the image analysis comes in, and also where the AI comes in that is trained to detect what part of that image is actually shoot. Where is the seed? Where is the main root? Where are lateral roots? And this is slowly turning the raw data into numbers, because then you can add up areas of these individual segments. You can detect maybe root tips or internodes on your root system. But this needs a lot of training. So if you talk about AI, you need to label and train the AI on what's happening. And just to illustrate it, this is a colleague of mine really annotating the data manually. So he is making information out of the data, and then the AI is trying to mimic that. So this is the labeling procedure that you need to go through to train the algorithm to do exactly what he has been doing manually, but then on thousands or 100,000s of images. Yeah, and because I've got no idea about your background, if you have a scene, so whatever, a blob, and you make a picture, you need some kind of illumination to turn that object into a digital version, actually, of this blob. And then because of the scene, the lens and everything, you get a certain representation on all the pixels in, for instance, your mobile phone. And your mobile phone then switches on or off certain pixels on that sensor. And then you get a certain representation of your object. And as you can see here, this is a high resolution real version of your scene. This is a much lower resolution, right? So it's a bit blobby, a bit blocky. And this is already the first step of losing information. And then algorithms need to make sense of this collection of numbers. So this, in this case, grayscale, so slightly higher values here, slightly lower values here. and 0 values over there, with maybe even some noise. To illustrate this further, so this is the imaging part, right? So if you have a random image, like also again from your mobile phone or from some kind of imaging system, and you zoom in a lot, then this is actually what the algorithms are working on. So here, maybe this is a plant, right, with leaves, maybe this is a stem, but then the algorithm is diving into each individual pixel, and he needs to figure out if this pixel belongs to the background or to the plant. And you can see actually that it's quite fuzzy. So there are quite some pixels that are slightly green or slightly blue. This is already challenge number 1. So what is part of background? What is part of foreground? You could for instance say, okay, all the pixels that are a bit green belong to the plant, and all the pixels that are quite blue belong to the background. But then somewhere you need to make a decision on to which it belongs. And this is actually image analysis. It's this thresholding. You could do it in 2D. Of course, you could also do it in 3D. The example that I will show actually also face this challenge. But with root phenotyping, the problem is even worse because you need to somehow make a picture of your root system. And it is difficult actually. To illustrate it, I would like to zoom into one of the climate rooms where the focus is on root phenotyping, with only a short side note on the shoot phenotyping that I would like to share. This is one of the buildings of the phenotyping center. The moment you enter here one day, you can freely walk around. So this is the top view perspective of an imaging system that looks at the shoots of, in this case, lettuce that grow under controlled conditions with certain LEDs in a certain temperature, with a certain nutrient mixture. Roots are hanging in the either rock wool or the ebb and flood system here, and they grow, and the cameras monitor how they evolve. And then these pixels need to be identified as part of plant or part of background, and that's why we have these black plates. And so we have a beautiful black picture, and whatever is green belongs to the plant. To illustrate it, it's quite easy to segment these images. That's why the plates are black. And then we can say, okay, these pixels belong to the leaves. All the other pixels belong to the background. And then we can analyze these pictures. A colleague of mine has been doing this. And he can even track leaves over time. But that's not the goal of today to explain. Let's dive into root phenotyping, because there the challenge is, how do you visualize your root? And we have been designing this for many years, also in alignment with Jülich. The experts at Jülich also had a similar rhizotron system, and we didn't want to reinvent the wheel. So what we do, we try to squeeze the root system into a 2D system with a glass plate, so we are able to look at the root while it is growing in a slightly tilted plate downwards along the glass plate, and we can see the seed over here on the top, and we can see the root going down here because it grows along this glass plate because the plate is tilted a bit. And that enables us to look at the root while it is growing in an artificial soil system. So that's another challenge. Is this representative for the behavior of your roots in the field? That's actually an answer that I cannot give you. And it will probably matter what kind of soil you put into this rhizotron, what kind of experiment you are validating. And that requires many experiments that we will do in the upcoming years. Okay. And then we design the system to have as many plates as possible inside one climate chamber to enable us to measure many plates instead of this one example from the X-ray, but more like 300 plates within a few hours without any human interference. But actually the human is the weakest link in the loop. So it's a small chamber, 4 by 5 meters, with 300 plates stacked here, tilted with a certain angle, and the shoot is growing above the plate, and the root is growing along the glass plate in the dark downwards. And then we can actually see the root growing as an imaging system takes these plates one by one from these lanes and scans it with a top view camera, a side view camera, and a root, basically a flatbed scanner. And this is actually the imaging device in which we take these images, and everything is completely automated. So this is expensive, very complicated, but from our end needed to objectively measure root performance differences between species, between genotypes. So another example, this is one of those plates, small tomatoes growing. If you look at the root, you can really see it by eye, so the camera can actually do the same thing. So one of the scans is an actual scan made by the system, and the resolution is quite okay. So you can see many small root details, but this is raw data. So we need to turn this into information by going through that process that I described. Okay, so which pixel actually belongs to a root, which pixel belongs to the soil. And then already you can see that some parts of the root are hardly visible by the human eye, by your eye, and other parts are obvious. And the roots are really hiding once in a while. So they can even be gone for a few centimeters and then pop up again. And then we need to guess where it came from. To illustrate it, this is a time lapse of one of the plants in the system. So you can see that the plant is growing and every day it gets slightly bigger from a shoot perspective, side view, top view, but also actually from a root perspective. So the plant is growing and the root is actually diving downwards. And then it's of course a matter of keeping track of the root system with image analysis, figuring out how fast and how deep it is growing, how many roots is it forming, and how does this specific plant compare to its competitors or its brothers and sisters. Good. So I would like to now go back to Mentimeter. Hopefully many of you found Mentimeter. Because yeah, exactly. This question. So if you are with me, please go to this slide and also fill it in. The more actually, the better. Root phenotyping tools are hardly there. There are only a few systems out there. And if you think about root phenotyping tools and cameras, then of course, often the answer is no, because it's very complicated. On the other hand, we also have a root washing facility here next door on the campus, where people simply dig out plants, they wash the roots, they monitor how big these roots are, they measure the weight of the roots, the fresh weight and the dry weight. And actually that is also root phenotyping. Many of you are potentially doing that on that level, but that's not automated phenotyping, but manual phenotyping. Okay, another question actually, this is one that is more interesting. There's data all over, right? So we are in the AI era at this moment. We need to deal with data. So this is exactly what I expected. So there are most of the researchers are using Excel or basic data analysis steps to make a graph, to compare some numbers, to look at your project budget, etc. A few use MATLAB. R is quite popular. Python is really quickly becoming the main development software language worldwide. And then there are many people that use all kinds of tools. And this is something already, if you want to team up in a project or with colleagues, a few of you work in MATLAB, others work in R, and others work in Python, you cannot exchange code. So that's already a dilemma. I actually promote that everybody is switching to Python, and then you can collaborate, exchange code, something that we will also do within PHENOWEX on drone data processing. Now, off topic for today. Yeah, let me see. Now the answer is more complicated. What are you doing if you don't use phenotyping tools? Majority, the next one is that you are using cheap sensors. So that's cell phones, cameras, Intel real sounds, Raspberry Pis. A few of you use drones, so that's good. And a small group is using some high quality sensors, so the more expensive ones. And only 2 of you have a large facility. And what is good to know that once in a while, in every country worldwide, the Ministry of Science or the Ministry of Agriculture announces a call for large-scale research infrastructure. And whenever this call is out there, which is within every country with a different pace and a different rhythm, and also with a different budget, you need to apply for it. And then also I or we can help you to write a good proposal to enable you to get a certain amount of money to buy your own facility or equipment. Cool, thanks for answering. I think that's it, and this is the discussion. So I can imagine that we cannot have a live audio connection. If you have ideas or questions, you can dump it here. And we can go back to these slides when I'm done in a few minutes. I would like to show you a few more things. But if you have some discussion topics or which tool is the most useful, please place it here while listening hopefully a bit to me in the meantime. So going back to the rhizotron, it is a very complicated, very advanced approach to measure roots, but I think it works for certain applications, right? So we can, and we try to be as generic as possible. So we can vary our soil types. We have an option to inject with a needle some fluids. We could add maybe specific bacteria or salt or biostimulants, or maybe even heavy metals. A colleague of mine actually did it the other way around. He actually sucked out fluids to analyze what's around the root. And we did it on as many plates as possible to allow GWAS to have a decent amount of control plants and treatment plants. There's a bit of the same information again, but slightly more detailed. So we have climate data that we can vary. We have soil type, but an experiment is quite short. So these roots grow down. Before you know it, actually they reach the bottom, and then the roots start crawling over each other, and your experiment is over. So an experiment is only 3 to 4 weeks. And if you find this interesting, I invite you to suggest a pilot experiment to how it works for you. There's at the moment even a colleague from Turkey working with us for 8 weeks as a guest researcher. There's a guest researcher from Japan at the moment with us for a year. And it's all meant to exchange ideas. There's a colleague from Italy at the moment with us working on the drone data. But yeah, to illustrate all the potentials, I'm mainly talking about the potential, but there are many pitfalls, things that can go wrong before you buy it, but also when you are using it. It has been described by my colleague Hendrik Porter. Many of us actually joined this publication to suggest all kinds of issues and potentials. And there's a specific section also on roots. And this is just a copy paste out of the publication. But it's always complicated with roots that you would like to see the roots interact with your experiment over time. So would you really like to see your root growing during your experiment? So that's where the rhizotrons could come in. On the other hand, you could also decide to only look at the roots at the end of the experiment, dig the plant out, look at the roots, and compare controls and treatments. So that's what we do in the greenhouse. Therefore, I'm going to add one more example actually, because often root phenotyping experiments are on drought. And that is something that we do constantly actually also in the greenhouse, where we do not look at the roots. Actually, this is a big trial on potato. This is the control table with ebb and flood. They are receiving water. So we pump water on the table. So all pots get an even amount of water. This is the treatment table. And we have inoculated these soils with fungi. And then we monitor how the roots interact with the fungi by simply looking at the shoot. And that is completely straightforward if you have the tools and the scale that we managed to purchase. So we take a top view picture of these plants. We know that the roots are interacting with the fungi, but actually you can see from a shoot perspective that the plants actually don't turn pale, or they don't turn weak, although this one might turn a bit pale, but actually the fungi is blocking the development of the plant. So the growth curve is flat. It's not completely flat, but it's very flat compared to the control table where the growth curve is quite steep. And the growth curve is generated by making a 3D scan of each individual pot. And then we simply add all the points together, which is a proxy for the fresh weight. That's it. So we have a lot of tools out there. So we scan all these plants and we estimate the total volume of your shoot on the top. We plot it over time. We select the ones that are in the treatment table while still being quite green and big over time. And those are the genotypes that were suitable for the next level, which is a field trial. And the same goes actually for a drought and salt experiment with quinoa. So here again, we can't see the root system, but we have introduced a certain salt mixture and a certain drought regime to individual pots. We've been doing this experiment for many years now. By looking at the weight change of each pot, we can see how the roots are using the available water in the pot over time. So this is the weight changes every minute measured by the scale, monitored by this small microcomputer. And for each individual day, each line is a pot. And we can see the weight change, basically the transpiration rate, peaking at the peak of the light level in the greenhouse, which is around lunch, starting in early morning, but also slowly falling down in the afternoon when the sun is setting, but also the plants are literally going to sleep. And you can also profile this water uptake behavior and compare them with each other. And in that way, basically objectively judge how these root systems actually interact with the soil and the available water. So it's not root phenotyping, but it's actually all driven by the root system. Also, this has been published already a few years ago. And also this has led to field trials. I think I have a picture in my slides also of the result in field trial. Actually, to be honest, not 100% sure. But it's all about data. So we capture data. So if you look at this scanner, he is taking these pictures of the individual pots one by one. These pictures are generated by a camera in the greenhouse on a local PC. This PC is standing in the greenhouse, so we would like to get it away there as quickly as possible before the hard drive fills up. So we are uploading it to, in our case, Azure. We do a bit of processing in Azure using Python. Then the users, so for instance, you or a customer can actually log into Azure to look at the images, to look also at the processed images. So the tables and the growth curves. But after a certain moment in time, a few months, we dump it into an archive because otherwise it gets simply too expensive to have it all in Azure. So we dump it on tape using iRods. And the tape archive is simply a facility that is on campus, quite scalable, super slow, but cheap. Okay. And on the other hand, this is again a bit of Python. We enable people to use the data and transform the data into these graphs. So this is this digital biomass graph from whatever experiment. That's actually irrelevant. This is a Python notebook that is on the one hand having code. So this is Python code, but also generates numbers, and users receive this Python notebook to play around with the raw data themselves. So they can change these graphs easily. They can mess around with whatever they want to because they always want to have something specific. And the Python notebook is easy to use and very interactively shared. It's basically a text. Even in Google Colab, for instance, you can run this Python notebook. If you have the CSV file, the images, you can do it on your local PC or on your cloud or within Google or within Azure. Again, a few more examples. See, there's somebody in the chat saying something. Oh, okay. Thanks. And we have been trying to go towards fields with this root phenotyping challenge. And we designed a very fancy and simple plot. This is the bucket sensor, as we call it within our group. This is cheap and it is scalable. It is basically a whole bunch of electrodes that are connected through these screws, and they are in contact with the soil. So inside the pot, there's nothing except the other end of the screw. And we measure the electrical impedance by quickly monitoring the current between all the different combinations that you can imagine between these screws. And if there's a bit of a blob of water in the soil, or some object that has a slightly different property than the rest of the soil, then you can actually reconstruct this. So we can reconstruct not the root, that's too small, but we can actually reconstruct a tuber that is growing on the root of this potato plant. And this actually worked amazingly well. So we were quite happy with it. And the electronics that we use here are extremely cheap. It's only a few €100 and you have a bucket sensor. But then it's only for tubers, not for roots. And we have been translating this to field situations. Another thing that is really accessible also for you, I saw that many of you use cheap sensors on mobile phones. It is simply an idea. So if you look at your mobile phone, it has a camera, right? So you can make a picture. But that's not where it should end. So we have actually new phones have high quality cameras, but also a little bit of depth registration. So if you take a picture of a box of tomatoes in this case, it knows the distance from the camera to the tomato. So you can also objectively measure the size of whatever is in your picture. And then we developed an app, which is a framework based on existing tools to turn that picture into measurements. And we have been doing this on tomatoes, but we are at the moment also doing this for roots. And just to illustrate it, so you take your picture with your own mobile phone, we structure the scene a bit, so the AI algorithm can easily identify the individual objects, in this case tomatoes. The AI can also detect if the tomato is upside down or straight facing the camera or sideways. And if he detected all this, he can extract trace, and extract diameter, look at the peduncle or the peduncle scar size, or the color. And this is the orientation analysis AI model, but basically a matter of training. And you generate numbers. And this is a very scalable tool that can be handed out to users all over the world, instead of going to our fancy facility where actually it's difficult to get there, and it's also difficult to constantly use these tools. This is for you ready to be used within weeks or maybe even days. So it's an app developed for variety testing of tomatoes in this case, and it just generates numbers. So you take a picture, generate numbers, maybe there's a QR code in the scene to list which metadata is relevant for these batches. And now exactly this is now done on root phenotyping, but then as you can't see the roots, you need to somehow take these plants out of the soil, put them isolated on, for instance, a black background. But if you have it, you can take a picture and the AI can analyze shoot characteristics, root characteristics, and it might help you to speed up your data collection process. That's the whole goal of this app. And these are just two examples of an application, but your mobile phone is an extremely powerful tool. Much more powerful than I think you are using it. So explore the potential. Okay, so I'm gonna sum up, and hopefully you have dumped some questions also in the last slide of Menti. Now this gap is my focus, especially to bridge the gap towards the biologists, the breeders, the plant experts to use the phenotyping tools. Hopefully today also made the gap slightly smaller. In the end, if we have managed to reach this situation, I'm also expecting that there will be many papers on, for instance, finding drought-resistant varieties, or that the food system has been more stable because of a tool, which is not completely relevant anymore, because the phenotyping system that is for me in this talk quite important is then just a tool in the process of growing plants, measuring your plants automatically, and reporting the outcome. But data is the burden, right? So we don't need more data, although we are drowning in data. We need to quickly extract numbers, graphs, information. And then the question is always, so what kind of information is useful? If you have a certain trait and it's not measured by the first version of the algorithm, then you need to develop more algorithms to extract your specific trait. Each experiment within our center is doing this. And what I'm promoting is actually that these tools, so these Python notebooks, for instance, are updated, and that people that find some cool algorithm to extract some kind of trait are giving it back to the centralized repository. And that the next user, which could be an MPEG user or somebody else, is reusing that code to not have 20 traits, but maybe 25 traits extracted. Yeah, and it's all about AI in the end. So I mentioned it a few times without any detail, but all these images can be analyzed with smart systems that extract information. And we have an enormous amount of data, also data sets available for people that would like to play around with the data because of the the developments within AI. So also outside of our community, people are in need of data to show the capabilities of AI, and we welcome them to use it. Okay, so I used a lot of material from colleagues and partners. I mentioned only a few names here, but let's go to Menti. Oh no, you can also jump in if you like.
Yes, thank you.
Yes. Not sure. Let me see if there's. Oh no, it's one slide. Yeah. Okay. Ah, yeah.
Rick. Yeah. Indeed. Thanks a lot, Rick, for your talk today. Indeed, the floor is open for questions, either via Menti or just by raising your hand and opening the microphone. I see there's some questions, so we can go via Menti and then place.
I can't see Zoom if I have this open, so then I can't see raised hands or anything.
Yeah, I will take care of the raised hands and I will stop the recording. Thank you very much, Rick, so far, and open for questions.